Welcome to the presentation on mitosis and the cell cycle. Mitosis is making a copy of a cell, an identical copy. Very much like a Xerox with a sheet of paper. Copy, identical copies. If everything works well with the DNA replication, you should have the exact copy of a skin cell, hair cell, any cell that divides by mitosis. Couple of mitosis definitions. Chromosome is one strand of supercoiled DNA and the proteins that support that DNA. Chromatid, the two copies of each chromosome. So after the S phase of the cell cycle, when DNA has replicated, there's two DNA chromosomes and they get stuck together at a centromere by chromatids. They get stuck together near the center, and that forms a chromatid. A diploid is a cell with a complete set of chromosomes. It's 2N. It's a normal regular cell. Almost all the cells in your body are diploid. They have two complete sets of homologous chromosomes. And a haploid is a cell with half a set of chromosomes. It's just listed as N. Haploid cells can also be called gametes or sex cells. They have one set of homologous chromosomes because the intent or the idea is they'll be joined with another. If it's an egg, it'll be joined with a pollen or a sperm cell, and each will add an N to the system to make a new 2N zygote. Reasons for mitosis. Cells can only get to be a certain size, so large. If it gets larger than that, the cell will split into two smaller cells because of a volume to surface area ratio. They just can't get bigger. They can't support themselves. Also, cells will divide rapidly in areas of high cell growth. That's skin cells, hair cells, tip cells. In other words, some of the cells, tips of plants, some of the cells are designed to reproduce rapidly. The life cycle of a eukaryotic cell is called cell cycle. The other important concept of this presentation. There are 5 stages to the cell cycle. The G1 phase, where the cell just is acting normally. If it's a skin cell, it's a skin cell. If it's a hair cell, it acts like a hair cell. Does whatever that cell does. In many cells, this is a great majority of their lives. Not all cells divide all that much. In most cases, when you're fully developed, nerve cells don't divide at all, or very, very, very slowly. So they would spend almost their whole existence as a G1 phase. The S phase is when the DNA replicates. This is the part where we have DNA replication. So the chromosomes are copying and becoming chromatids. The G2 phase is the second growth phase, where we're getting ready for cell division. So we need more organelles. We need to get ready. We need to double our number of mitochondria, double our number of ribosomes and endoplasmic reticulum. So when we do divide, each new cell, each half, will have enough stuff to get by. Then the M phase is where the nuclear division takes place. The mitosis phase, it's called. That's where the nucleus divides into two. And the C phase is the cytoplasm division, where the cell actually divides and we end up with 2 cells. G1, it's the longest phase in the cell cycle. As I said, it's where normal growth and activity takes place. It could be a very majority of the time that the cell is a cell, if the cell doesn't divide very much. If it's a hair cell or a skin cell, it's smaller because they do a lot of dividing. So it depends on the type of cell. Remember, at one point you were a single cell, so then through mitosis you divided and divided and divided and eventually became a human being. The S phase is starts the cell on its journey towards cell division. The first thing we have to do is copy the commands, copy the nerve center. So the S, the DNA, that's what controls the cell. So we have to copy the DNA, double it, replicate it, I should say, the DNA, so it's ready to a complete set to go to each new cell. G2 is cytoplasmic duplication. This is the phase where the organelles increase so that, again, as I said, getting ready for cell division. We need enough, if there's not enough mitochondria in each new cell, it'll die from lack of the energy of life, ATP. So we have to make sure there's enough mitochondria, enough lysosomes, enough of all the organelles, that when we do our cell division, actually, that there's enough of those organelles in each new cell. Just what I said. So the G2 makes sure this does not happen. Then we get into the M phase, and this is, a little bit more complicated because it has some steps to it. The M phase of the cell cycle, you can see most of the, it's a smaller phase, but it's very important. At the end of the M phase, you're going to have a single cell with two identical nuclei, and that's kind of strange, but shortly thereafter the cell will divide and you'll have two cells with identical nuclei, and that's back to normal. But this is kind of a interesting period. It has four parts: prophase, metaphase, anaphase, and telophase. These are the four steps or parts of the M phase of the cell cycle, and called mitosis phase. Prophase, the first step. In prophase, the chromosomes shorten, thicken, fatten, and become visible. So all this other time, they're all like a long string of chromosomes, a long string of DNA. But now as we get ready to do cell division, nuclear division, the chromosomes shorten, fatten, thicken, and in a light microscope, they can become visible. The little X's that we sometimes have grown to see and think that's what chromosomes are. It's actually two chromosomes bonded together at some central point. Two strands of DNA, two identical strands of DNA. So we've got we've got a larger and a smaller red X up there. That's the big red X is two strands of DNA held together. The nuclear membrane begins to dissolve, so we start to lose the integrity of the nucleus, and spindle fibers from the centromeres start to form, and the centromeres are moving to the poles. So we get these little rope-like things appearing. The M phase, the spindle fibers have moved to the poles, and the chromatids are grabbed by these centromeres and moved and lined up all in a line at the equator, at the center. And remember, each of those sides of the X, the big red X, the big blue X, little blue X, the little red X, each of those are exact copies of each other. So if they get pulled apart, we're going to pull the identical DNA strands to opposite sides of the cell. But this is the M phase, the middle, the line-up phase. Anaphase, the spindle fibers begin to shorten and they pull the identical chromatids apart. So each chromatid, chromosome, strand of DNA is being dragged to opposite sides of the cell, to the opposite poles, getting ready to form a new identical nucleus. So we copy the DNA and now we're separating it. This is a key part to the cell division, to make sure that you keep the integrity of the nucleus in each new cell to be identical. Telophase is the fourth step in the M phase. It's basically the opposite of prophase. Chromatids lengthen and disappear. They shorten, fatten, thicken in prophase, but here they lengthen and get skinnier so we can't see them. The nuclear membrane reforms, so we're getting two new nuclei forming with identical sets of chromosomes in each, and the spindle fibers begin to disappear. So what we're basically doing in a single cell is forming two new identical nuclei. We've copied the nucleus, we've replicated the nucleus, and now we're going to, the final step is to split the cell. The C phase, cytokinesis, is we have a cell with two nuclei in it, two identical nuclei in it. As I said, kind of an unusual situation. So it's time to divide the cytoplasm, that's what the C stands for, cytokinesis, cytoplasm division, and finish the step of cell division. An animal cell will cleave down the middle, kind of like if you took a water balloon that wasn't overly inflated and just pinched in the middle, and you'd get two balls. Of course, we can't separate them or they would burst, but that's close analogies we can get. And a plant cell will just simply lay down a new cell wall in between the two nuclei, and you'll have two smaller plant plant cells afterwards. Of course, the animal cells would be smaller too, but then they can get into the G1 phase, start to grow, and the cycle repeats. Your cytokinesis in a plant cell, you start to form these member-bound vesicles down the middle, and before you know it, we have two cell walls where there was none. I mean, we have one cell wall where there was none, but we have two cells now. We have two identical plant cells. Okay, and now again, whether it's a plant cell or an animal cell, we have two identical cells from an original, perfect copy. That's the idea of mitosis with a T, split. That's how I remember it. Mitosis, split, copy of the original cell.